So I've just got a couple of slides here really as an introduction to what we're going to do with the code. Luciano kind of already introduced the ideas pretty well, I think. But basically when we're talking about Genotype Environment Association analyses, we're interested in how has the environment shaped patterns of genetic diversity that you might see in your group of interest. And so there's a lot of different methods that Luciano sort of skimmed over a few of them. Today we're going to be talking about redundancy analysis, and we're going to use a data set that came from a paper in 2018 with Murray River rainbow fish in obviously the Murray Darling Basin. For those not familiar with the species, rainbow fish, kind of that size and smaller. They have a sort of a moderate capacity for dispersal. They don't tend to move a real lot. This particular species is the most southern or most temperate species in the group of Australian rainbow fishes. And they tend to get less abundant in the south as it gets cooler. And so looking at the patterns of aridity across the Murray Darling Basin, you sort of see the cooler, wetter areas in the uplands right down there in the south, and then some really quite arid areas as you go sort of further to the north and to the west. And there's sort of, if you head right up into the northeast, then you're starting to get into some sort of almost subtropical climates as well. So there's a range of different climates within the basin. And so here we're kind of looking to see, well, are any of these populations potentially locally adapted to that climate in their particular region that they inhabit. This is just from the paper, just to give you an idea about how the environment might vary across the basin and where the sampling locations that we're going to be looking at today are. So the Murray Darling, basically you've got the Murray River, which goes through sort of the border between Victoria and New South Wales for a lot of its course. And then the other major river is the Darling River, which comes down from that northeastern sort of end of the basin. And it's a really heavily modified environment, lots of in-stream barriers and things. There's been some progress in putting in place fish ladders and other ways to allow fish to move around the basin, but they're still probably less able to do so now than they were in the fairly recent past. And just to give you an idea about what the kind of basic population structure in the system looks like, this was probably admixture. And basically we can see there's essentially 6 different genetic clusters there that are reasonably well spatially aligned with the river network, which is what you tend to see in like a dendritic river system like the Murray. And Luciano really went through what RDA is as much as I was going to. Basically, we're talking about ordination analysis here. You can have an unconstrained ordination where you're just looking for the main axes of variation in your data. Constrained is where you take those main axes and then fit them into combinations of another set of predictive variables. So in this case, it's our environment, and examples of that are CCA and RDA that we're going to be using today, but we don't really need to. We can talk about that later if people have got questions. And yeah, this was just showing what the model actually is. You're looking to see how the variance in one set of data might explain variance in another set. So in this time, how the environment might explain variation in or variance in our genotypes. In the case of when you've got population structure, you might get patterns within your genotypes that vary because of their relationship in space that might be independent of variation in the environment. However, they also may co-vary with the environment. So it does get complicated, but you can do what's called a partial RDA, where you're not only using the first part of that model, but you're also partialing out the variance that can be explained by something like space in this case. So the relationship between the locations of your samples, you can get rid of the variation that is in your genetic data that's associated with that, and then try and narrow down or sort of home in on the parts of your genetic data that are explained by the environment. And there's an almost limitless way that you might be able to model how population structure might impact your data depending on your system. We'll talk about a little bit of that in a minute too. And that was just, yeah, another example. And just quickly before we get started, I think there's some fairly accomplished coders in the audience here. I'm not one of those people. Use whatever works is kind of my, I don't know, philosophy might be too strong a word, but my what I tend to do. There's a whole range of ways to do anything you want to do in R. I learned or started learning R maybe 10 years ago using base R. As you can see, depending on what you want to do, there's a whole range of different, I don't know, universes, I guess they call them, that to complete pretty common tasks. One of the things I really thought was good yesterday in the talk about reproducibility was making your code readable, not only for other people using it, but even for yourself when you go back and you go, what the hell did I do here? A quick example, a for loop. If you go onto Stack Overflow trying to figure out why your for loop isn't working, you'll find all sorts of criticisms and that it's not a very R way of doing things, and you should be using lapply or these days even map. That's fine. If I'm reading quickly through some code, I find it much easier to read what I'm doing in a for loop than having to go through my lapply line of code. So what it looks like for me, these are usually what I do. There's nothing to stop you using any of the others. They're all supposedly more efficient. Do whatever works. That's the takeaway message. So let's have a look. Where did we get up to? Yes. Okay, we're getting down there. And all right, yeah, it's fairly large here. So basically the data for this is taken from that Rambofish paper I pointed out. It's a much smaller subset of the whole data. And I kind of modified it a little bit to highlight some of the bits that we're going to be looking at here. So it's not actually a real representation of what's going on with the Rambofish, but it's pretty close anyway. And so if you can run this data preparation chunk, I'll get that running now. Basically what that's doing, it's loading your R data, which is a Genotype object. And then if we swap to here, it's easier to see. Now, the thing I wanted to point out just briefly here for people not familiar with the way these things work, if you look at the population slot for the Genotype object, we've got the population code for each individual in a vector at the top, and then you have the levels, so which populations are actually in the data at the bottom. You notice one thing, they're not in the same order, and that can catch you out sometimes if you're not looking. By default, structures the levels in alphabetical order, whereas your data might be in some type of geographical order that kind of makes sense to use. So if you want to reorder it, then the next line of code there is just reordering the levels in that object. And so eventually, scroll down and do it here. After running that, you can see that the levels are then in the same order as our data. You don't need to do that as long as you're aware of the way it works, but I tend to do it because I will quite often forget and then wonder why my plots are all weird and not doing what I think they're doing. And so if we have a look at the, we've converted it to a gen_ind object. I tend to work with those almost a little bit more than gen_light, where I can actually look at the data a bit easier than with a gen_light object, particularly the genotypes, the tab slot, and it's useful for converting into a whole range of other things. So I've got a package called melfoo_r, which is mainly just to keep my own kind of useful functions somewhere that I can find them, and for the lab to use as well, but it's open source on my GitHub. And a lot of the things, they're just wrappers to take a gen_light or gen_ind object and then run some other analysis without having to figure out how to reformat the data, and it'll do all that automatically. So I tend to use gen_ind objects for that because I find it a bit easier to wrangle. And if I come back to here now. And so, yeah, we can see that we've got 249 individuals, we've got a subset, 5,000 SNPs, and 10,000 alleles for those SNPs. Okay, so the first sort of discussion point or decision we need to make is that ordination analyses typically don't handle missing data either at all or very well. And so you've got to decide what we're going to do when there's missing data in your genotypes. So you can just remove any of the SNPs that have missing data. That might leave you with a fairly small data set. You can remove individuals with missing data. Most often, there'll be a little bit of missing data spread throughout most, if not all, individuals. And the third option is to impute the missing data. So you take a SNP in a certain individual and figure out what the most likely or what a reasonable data point to insert where that missing data is might be. So there's kind of, well, three options I've put here. There's probably more than that. The most common genotype across all of your data, depending on your study system, that might be okay. If it's one fairly panmictic population, that might be a good and simple option. It also is a fairly conservative option because with the GEA analysis, you're trying to find differences among individuals or populations that might be related to the environment. Now, if you're going to impute some data and have that imputation actually affect the results, by inserting the most common genotype across all your data, you're making all your individuals or all your samples look more similar to each other. So it's sort of a conservative way of doing it, that you're not going to increase necessarily the false positives. You might end up with more false negatives that way. You need to think about that. The most common or most likely genotype per population, that might be a reasonable way to go if you've got a more structured data set. And what we're going to do today is, like I said before, is essentially just a wrapper function that works around the LEA. R package has a function that takes, it runs a SNMF that Renee went through, I think, the other day, and I've forgotten what that stands for now, but it's essentially like a structure analysis, but much faster. It'll split your samples up into reasonable groups. Excellent. So if you don't want to worried about this, then go and see Arthur. Yeah, there's another option there. Okay, I didn't realise that. So, all right, that's cool. Data has an option to do that as well. And so it looks at the allele frequencies for each one of the inferred populations and picks the alleles or the genotypes to impute based on where your sample fits in to those to that structure. And you of course need to know, you need to have some sort of hypothesis as to how many populations you actually have. So we saw in the introduction that we had already run that. We knew that k equals 6. So if we run that, you'll end up with a data set that no longer, it'll just take a couple of minutes. So we had about 1%, I think, missing data before, something like that. And we're going to end up with a data set with no missing data. Yeah, so the question was just about how, in this case, we decided that the K value, where's my pointer gone? The K value here was 6, and yeah, that's from previously running admixture and doing some other population structure analyses. You can, if you want, modify this code so that it can find that most likely based on cross-validation for you. Most of the time, by the time you're getting to this point, you've already got a fairly good idea about sort of how your data might be structured already. Okay, so, and now on that bottom line, we can see that we now have no more missing data. I'm going to try and speed up a little bit here. And so there's another step here that you absolutely don't need to worry about doing. I tend to get a little kind of fussy about this sort of thing and just like to be neat. Depending on how your data was generated, it might not necessarily be ordered from major allele, minor allele, major allele, minor allele. Sometimes there's good reasons to have it ordered like that if you're interested in plotting the direction that different populations might be changing over time, something like that. There's a sort alleles function in the utils .r folder that will just fix the order of the alleles in your genend object to be ordered from major, minor across each locus. So if you had run the function to sort the order, you'll notice here the first SNP, the first column is a C, the second one is the G, but you can see even in this little sample of the individuals here, the major allele is actually the G, and you can see after the sorted one now we've swapped the G and the C around, so that's ordered major, minor. Again, optional, it keeps things neat though. All right, now, there's a note there about the function, we can talk about that later. You have a choice to make whether you want to run your RDA on an individual basis or on a population basis. If we want to do individual based, we can format our data so that it's ready for the RDA by taking the data from the tab slot, which is just your rows of individuals by each allele for each SNP, and because that half of that data is redundant, it's an allele count, so if you have a 0 for allele 1, that means you have a 2 in the second allele. If it's a 1, the other one's a 1. If it's a 2, the other one's a 0. So we can get rid of every second column. This little bit of code just does that, and then it tidies up the locus names. These are just example kind of data numbered SNP 1 to 2, but whatever your locus names are, it will clean that up. So if you go ahead and run that code, if you're doing it in terms of the population, that's what we're going to be running today. It kind of makes sense with the way the fish are sampled here, that we go to one location, sample 20 or 30 fish, go to another location, 20 or 30 fish, and do that. At a population level, you want to base it on allele frequencies is the easiest way to do it. And you can run the next lot of code. Okay, and if you run the population level chunk, you'll see that you end up with a similar looking matrix, but this time instead of allele counts for each SNP and each individual, you have each population or each site is a row, and then each SNP gives you the allele frequency that you find in that site.
I just missed why you would do by individual or by population? Like,
I'm just trying to show, as an example, you can run the RDA using individual genotypes or population allele frequencies, and really depending on your type of data and what you're trying to achieve.
But does that depend on your question?
A little bit, and probably on your sampling as well. So if you have, you know, maybe a species that you've sampled kind of more sort of, you know, each individual is sampled in a different location kind of thing, then it might make sense to be able to keep all of this separated by individual. In this case with the fish, we've gone to, you know, one site, and 20 of our samples come from that one spot, and then the next site, another 20 come from that spot. So in that case, you can still run it at an individual level, but I don't think you're really gaining a lot by doing that. So you can just use allele frequencies to do that. Depends how much structure there is in your data as well.
So if you have sampled individuals on a continuous, basically, way, you would go by individual?
Probably individual is a better way to do that then, yeah. Okay, so if we want to find out how our genotypes might be related to the environment, we need some environmental data. And so there are a huge number of places we can go to find that, and things to think about here. We're really pushed for time now, so I'm just going to kind of skim over these a little bit. Today we're going to just be downloading a couple of variables from WorldClim, and we're going to just format them within the R environment to use in our RDA. So it's just a sort of a sample bit of code on how you can go about downloading some data and running the analysis with it. There's a couple of options you need to set here. One is just telling it where it needs to download the data to. And the other one here, you don't always need, but sometimes connecting to this particular server takes a long time out, so you're just increasing the amount of time that R is prepared to wait for a server connection before it tells you that it's no good. So you can run those, and then you can sort of explore which environmental variables are available in this particular data set. This is using the STM predictors R package. There's a whole range. Today we're just going to look at the WorldClim simple variables. This, again, you can download the details about each layer, save it as a CSV, and you can look at that to sort of make decisions about what you might want to use. Add some colours to it. You can add whatever other things you want here. I've just downloaded a fairly hastily cobbled together main river system in the Murray Darling Basin, and then you can plot that, and you can get an idea about what your data actually looks like. Here, because we've downloaded multiple layers, if you run this whole chunk of code here, you'll get a PDF in your data folder that has each one of those layers listed in it. And then finally, this chunk of code here about extracting the environmental data. This is where you're getting your sample coordinates, and you're finding out what the environment at each one of those layers is, what the data is at that point, and you're putting that into a data frame for you to use in the analysis. And so we're going to call that env.site. I tend to do this with the analysis. I make a data frame, and each bit of relevant sort of information I just add to that data frame as I go. Then at the end I can just download that as a CSV, and that can be your metadata table that you put in your you know, Dryad or whatever when you publish it, or for you to reference back later. It just keeps everything all together. And so if you run that, you end up with your environmental data as well as the coordinates that we already downloaded, and there's a set of spatial vectors here that we're going to use to control for space that I'll talk about in a minute too. This line here, it's really good practice to put them every so often in your R code. Basically, you're just checking that the sites in that table, then that data table of all your metadata, your environmental data, are in the same order as your genomic data. In this case, it's the row names of the allele frequency table. So sometimes when you're doing these things, you can get them out of order. I will often love to put things alphabetically when you don't want them alphabetically. This just tells it to give you a little warning if the rows aren't in the same order in your two sets of data that you're analysing together. So you can modify that, and I use that all the time just to make sure that everything's doing what you think it's doing. A quick PCA, you can actually use the RDA function from vegan to do that. If you don't list any constraining variables, then the model just becomes a PCA. Plot that, it's not that impressive. You can add some colours, the shape, the marker types, some labelling, some labels for your axes, and then we can plot it again. So after we've done that, we're interested in using our environmental variables. Which ones do we use? It pays to have some type of hypothesis here that you can at least start with some idea about what you think is important. After that though, an RDA incorporates a regression analysis. If you have variables, predictor variables that co-vary, then that can introduce additional variance into your model that you don't want. So you need to try and at least eliminate the one of a set of variables that are highly correlated. So this is what we're going to have a look at here. You can just do a quick visual look to start with. This is just a correlation details.r script, and this just uses variance inflation factor analysis to look at variables that actually do increase the variance in the model more than the others, and it just uses a backwards I've forgotten what the term is for that now. What am I thinking of here? Backwards selection procedure to eliminate the one with the height that increases the variance the most, and then it'll test it again. Here we can see again, like we saw with the correlation, Bio1 seems to be the major problem here. Remove that. After that, all of these other ones were below a somewhat arbitrary threshold. People use 2, people use 10. There's a ton of literature about why it probably doesn't matter too much. We're not going to go into that here. So we dropped one of them, we're left with those 3 basically. And there's a little bit of formatting there to get it into a data frame that we can use, and just rechecking the correlations. Now that looks pretty good. And we already talked about this, about the need to control for spatial structure or not. There's a ton of different methods to do that. That's probably fairly dependent on your system. What we're going to do here is use something called an allele frequency covariance matrix, or if anyone's familiar with the software Baypass, it's their omega value, and that's basically accounting for the sort of similarities between your populations in the genetic data. So it's accounting for the structure, I guess related to drift is what it's supposed to be modelling. So if you, it's not said there, I don't know, if you look at Gates et al., which is in the document there, another lab member, Katie Gates, another postdoc in the lab, she's got a nice paper in Hereditary which uses this method if you want to learn more about that. Okay, some more formatting. So these 2, omega X, omega Y, are the omega values. Then we can run our RDA. So we've got the allele frequencies, we've got all of our retained environmental variables, CSV of that, and here we end up with the vast majority with Bio 15, which is seasonality of rainfall, I think, from memory, coldest temperature minimum temperature in the coldest quarter, and annual precipitation, I think, is the third one there. You can then use those candidate loci to run an RDA at the individual level if you want to see how what it looks like within each one of your populations, whether there's some that might be spread more out, because basically what you're seeing here at the population level is kind of an average. So you've got one marker for each one of those sites, but there's multiple individuals within those sites. So you know, this one here is the position of the average individual, or the allele frequency in that site. There might be some very different individuals in there that don't actually fit that pattern very well. So if we run, we can reformat the data into an individual level. We'll get down to the plot here, and the plot doesn't show up. Hey, there we go.
But yeah, we have the last plot.
All right, and that was a little bit rushed in the end, so please come and find me and, yeah, ask why it's not working. I'll try and figure that out. All right, thanks, guys.